Okay then, Year 2. So this next lecture on the OV 2023 Visual Perception module discusses the visual field representations that you find in the primary visual system. And actually this is extremely important from the perspective of clinical optometry, because damage to the primary visual system always results in the loss of visual perception in a specific part of the patient's field of view. And in your future lives as professional optometrists, you're going to spend quite a lot of time putting patients that you suspect may have a visual field loss into automated perimeters like the Humphrey Field Analyzer, after which you'll be invited, to diagnose the location of the lesion. And it's also extremely important enough that we're going to be doing a tutorial this week, which is focused very much on the visual field representations in the primary system. Okay, so by way of a reminder, I've told you this before, but it's worth repeating. By definition, the primary visual pathway, which runs through the primary visual system, comprises the ganglion cells of the retina and the axons that they give rise to, which travels from the eye down the optic nerve, passes through this X-shaped region at the base of the brain called the optic chiasm, and then those axons enter into the optic tract, and then the vast majority of them end up in the lateral geniculate nucleus of the thalamus. There they activate neurons in the LGN, and there are many millions of LGN neurons, and they give rise to axons of their own, which initially exit the lateral geniculate nucleus, pass into a white matter pathway called the internal capsule before entering this big chunk of white matter here called the optic radiations, the destination of which is the primary visual cortex, or V1 cortex, in the occipital lobe, again where they make connections with particular neurons in primary V1 cortex. Now, one of the things that I've tried to emphasize before, and again is worth repeating, is that you have to realize that your visual field, as indicated here by these sort of greenish and yellow structures, your entire visual field is split down the middle by a vertical line which is known, as shown here in red, as the vertical meridian, and it splits the left half of your visual field from your right half of your visual field, so that you effectively have two separate hemifields. And an important point, of course, is that via the optic pathways, the left half of your visual field becomes represented in the right side of your brain, and vice versa. The right half of the visual field becomes represented in the left side of your brain. And this is because nasal axons from each of the two eyes cross the midline when they reach the optic chiasm, whereas temporal axons remain uncrossed, they remain on the same side. And I'll remind you about that arrangement in just a little while. But a significance of this is that damage, for example, to the right primary visual cortex will therefore cause loss of vision in the left half of the visual field, a condition which is known as hemianopia. In fact, as we'll see, damage to the primary visual pathway at any point, if it's complete, at any point after the optic chiasm leads to a hemianopia affecting the opposite half of your visual field. Okay, so the sort of main objectives of this lecture is to talk a little bit more about the primary visual pathways, to emphasize the main message that all of these pathways are organized in the form of retinotopic or visuotopic maps, actually with large territories of those maps devoted to representing the fovea or macula, that is to say, to over-representing those parts of the retina, these being, of course, the central high acuity parts of your visual system. In the first part of the lecture, we're going to look at how retinal axons travel down the first part of the pathway, which is the optic pathway. They do change their retinotopic order a bit in the nerve. I'm just going to mention that in passing. It isn't really of super important clinical significance. We're going to talk about the line of decussation, the line that splits your retina into a nasal and temporal half, from which the ganglion cells of the nasal retina cross, whereas the temporal axons remain uncrossed at the chiasm, and we'll review the significance of that and how it leads to the generation of maps of the opposite hemifield after the optic chiasm, for example, in the lateral geniculate nucleus and in the primary visual cortex. And then the final part of the lecture, we'll look at the pathway from the lateral geniculate nucleus to the visual cortex. It's also known as the geniculocortical system. So we'll follow the axons of the lateral geniculate axons as they enter the optic radiations. And there's a really kind of important and sort of main message to get across in that last section of the lecture, which is actually that there's another division within the optic radiations whereby the inferior and superior, that is to say lower or upper visual quadrants, or representations of the lower and upper visual quadrants, take different routes through the optic chiasm. Through the optic radiations. One of those routes is very direct, but another one takes a rather loopy pathway to reach the primary visual cortex. And we'll look also finally at the detailed map of the opposite hemifield in V1. Okay, so that's the sort of general plan and some of the main messages I hope to get across. But let's begin at the beginning with the retina. As you can see, this picture on the right is a normal fundus image. You can see the location of the asterisk here is pretty much where you'd find the fovea, with the circle around it showing the immediate sort of parafoveal region. And the first point that I wanted to make is that both the fovea, the parafoveal region, and in fact the region that surrounds all of that, which is kind of this area here, which is known as the macula, are all quite small parts of the human retina. So, for example, the macula, which would be in this sort of general vicinity here, is only about 5 millimeters in diameter, and that equates to about 15 degrees of the center of your visual field. In broad terms, 1 millimeter on the retina is something like 3 degrees, something like. The fovea centralis, the little pit that you find here at the location of the asterisk, that's only about 1.5 millimeters in diameter, which would correspond to just the central 3 degrees of your field of view. The central 3 degrees, again, sort of a ballpark. If you put your arm out at your arm out and stick your thumb out, and you're at arm's length, your thumbnail is pretty much about 3 degrees of visual angle, corresponding to what you can see with your fovea centralis. So these regions of the retina, the fovea and the macula, are both pretty small and are only devoted to a small part of your central field of view. But these regions of your retina, just around the fovea and within the macula, actually contain something like 66 to 75% of all of the ganglion cells of your retina. As a consequence of that, your optic nerve, the axons that run from the eye into your optic nerve, around about 66 to 75% of them have come from the ganglion cells in the fovea and retina. So the representation of the fovea and retina is already overrepresented in your optic nerve, with 66 to 75% of the axons devoted to processing information from central vision. And this overrepresentation of central vision is then simply maintained all the way through the primary visual system, as a consequence of which between 66 and 75% of the territory in both your lateral geniculate nucleus and within your primary visual cortex are also devoted to processing information from these central retinal regions. Just a couple of other things to talk about in relation to the fundus image here. I've shown you this thing here, this dotted line, which corresponds to the line of decussation. The word decussation means to cross the midline. So this dotted line actually corresponds to the visual projection of the vertical meridian, that line that splits your visual field into a left and a right half. As projected onto the surface of your retina, it forms this vertical line that perfectly bisects the fovea and splits your retina into a temporal half on this side and to a nasal half on that side. The nasal half, of course, actually also including the optic disc. So these are the temporal nasal halves as related to visual field representations. And one of the things, of course, that you can see in the fundus image are these blood vessels. In fact, what they are mainly are branches of the central retinal artery entering here on the nasal side of the optic disc and fanning out over the retinal surface. And of course, as you can also see here, most of these blood vessels of the CRA, those running into the nasal retina, those running superiorly and those running inferiorly, take a fairly sort of direct and straight path across the retinal surface, whereas those that are going to the more peripheral parts of your temporal retina do that sort of classic arcing or arcuate pattern around the fovea and macula in order to make sure that the fovea centralis in particular doesn't have blood vessels running over it, which would cause light scatter and hence reduce the visual resolution of that region. So that's the reason, why the fovea centralis is an avascular, at least from the central retinal artery point of view, part of your retinal surface. Okay, now you have on average something of the order of 1 to 1.5 million retinal ganglion cells in each of your eyes. And the axons of those retinal ganglion cells run towards the optic disc or optic nerve head in the NFL, which stands for the nerve fibre layer, on the inner surface of the retina right next to the vitreous. And as the words on this panel say, the axons that are running over the nasal, superior and inferior parts of the retina take fairly direct and straight routes to exit the eye down the optic disc or optic nerve head, whereas those from more peripheral parts of the temporal retina take an arcuate course, kind of in the opposite direction to what we saw for the blood vessels doing, whereas those that come directly from the fovea and from the surrounding regions of macula are bundled together and leave the eye as a separate group of axons, which is known as the papillomacular bundle, and that's the bundle that contains about 66 to 75% of the axons leaving the eye. And this picture is intended to illustrate at that point and how it is that within your optic nerve there is a map of the retina. So this view here of the retinal surface is meant to be kind of looking at it head on, a little bit like you do in a fundus image. But now instead of looking at the blood vessels, we're looking at the bundles of axons that are leaving different parts of the retina here at the optic nerve head. And so each of these kind of streaks that you see moving towards the optic nerve head are meant to be bundles of ganglion cell axons that have come from different parts of the retina. And so you can see the bundles of axons coming from nasal, superior and inferior parts of the retina are travelling fairly directly. Those from the temporal periphery are taking this arcuate course around the central fovea, whereas those coming from the fovea and macula are grouped together in this thing here called the papillomacular bundle. But there's more to it than that, because the axons, as they leave the eye, are not just coming from the nasal, superior, inferior, and temporal retinal quadrants in a fairly orderly way, but they're also maintaining their centre to periphery positions in each of those 4 retinal quadrants, and that's what I've tried to illustrate here on the right side of the panel. So this is meant to be sort of part of the nasal retina over here. And each of these things here is a retinal ganglion cell body with its little dendrites sticking out. And the line that you see coming out of each of those ganglion cell bodies is the axon that is heading towards the optic nerve head and then afterwards heading towards the brain. And the point that I'm trying to illustrate here is that here we've got a retinal ganglion cell here in the nasal retina out towards the periphery, and here its axons travelling along the nerve fibre layer, and it dives into the optic nerve head at quite a peripheral location right at the edge here. This next nasal ganglion cell, which is kind of halfway along the nasal retina, also has an axon travelling in the NFL, and it leaves the eye at the optic nerve head next to these peripheral ones. And here's a more centrally placed, that would be a ganglion cell somewhere around here in the nasal retina, sending its axon into the optic nerve head, but now in a more central position, somewhere around here in the optic nerve head. So the ganglion cells from peripheral to central along the nasal retina have axons that leave the eye and enter into the optic nerve head in a kind of peripheral to central gradient. So that the map of centre to periphery in the retina, or across the retinal surface, is maintained in the organisation of their axons as they leave from peripheral to central in the optic nerve head. And the same applies for the superior and inferior and temporal retinas as well. So this is the precise retinotopic order of ganglion cell axons as they travel along the optic pathway. Here in the nerve, here as they pass through the chiasm, and here into the optic tract before ending up in the lateral geniculate nucleus here. I don't really want you to know the details of this. There were just a couple of sort of major points that I wanted to get across. These diagrams here, 1 to 4, are kind of the brain's eye view, if you like, of the optic nerve and the optic pathways. So they're kind of looking at the optic pathways from the brain's perspective. So one thing to point out then you see here is this is a cross-section. This is meant to be a cross-section through the optic nerve just as the axons have left the eye. And you can see there's an organisation of nasal ganglion cells here and temporals over here. But here in the indicated in blue is a kind of tongue of a large number of axons which have been derived from the papillomacular bundle. So in fact, just immediately behind the eye, the ganglion cell axons are not precisely retinotopically organised. The papillomacular bundle axons end up in the centre of the optic nerve a bit further down, where there is a proper kind of retinotopic order. And that order is broadly maintained through the chiasm and into the optic tract, although in fact, as it turns out, the fovea and macular axons tend to migrate a little bit dorsally within the optic tract. But here's a much more important thing, and it becomes of significance later when we get onto some visual field losses, is that down here at the very base of the optic chiasm, and the pituitary gland would sit underneath here, is where you find the inferior nasal quadrant axons. That's what NI stands for. So the axons that have come from the nasal retina, inferiorly in the nasal retina, are at the base of the chiasm, in fact just above the pituitary gland. Okay, so the optic axons, they travel down the nerve and then they arrive at this midline region, the optic chiasm, which is shown here in a real piece of anatomy, a photograph of the underside of a brain. Number 14 here are the final stumps of the left and right optic nerves. This X-shaped region is the optic chiasm, as indicated by the asterisk. Number 7 is actually the hypothalamus, which sits in your brain directly above the optic chiasm. And of course, at the optic chiasm is where this partial decussation, this partial crossing of ganglion cell axons occurs. In particular, it's the axons that have come from the nasal halves of both of your retina that cross the midline in the chiasm, and those from the temporal retina that remain uncrossed. So as a consequence of that, the right half of your visual field, or the right hemifield shown here in black, becomes represented here in the left side of your brain, and the left half of your visual field, as shown in white, becomes represented in the right side of your brain. And the reason for this is as indicated here in the diagram. So here is the nasal retina of your right eye. Okay, this is the right half of your nasal retina, and as you can see from the diagram, the nasal retina of your right eye looks at the right half of your visual field. That's because of optics. You know, the visual field from left to right becomes reversed across the surface of your retina because of optics. So the nasal half of your right eye is looking at the right half of the visual field, and so is the temporal half of your left eye, is also looking at the right half of your visual field. Because the nasal axons of the right eye cross the midline at the optic chiasm and report information to the lateral geniculate on the left, and because the temporal retinal ganglion cells of your left eye, which are also looking at the right half of the visual field, don't cross the midline at the optic chiasm but remain on the same side and also report information to the lateral geniculate nucleus on the left. As a consequence of that, the right half of your visual field, as seen by the right and left eyes, is represented in the left side of your brain. And of course, vice versa for for the left hemifield. And the significance of all that is that axon damage at different locations along the optic pathway will cause different visual field losses, which we'll come on to fairly shortly. But before we get there, I just wanted to point out that actually the optic chiasm is quite a crowded region. Surrounding your optic chiasm, there are a number of other neural and non-neural structures, all of which have the capacity to develop pathologies, and those pathologies can cause damage to the axons in different parts of the chiasm. So first of all, we're looking here on the left at essentially a kind of sagittal view of the optic chiasm. Here's meant to be the stump of one of the optic nerves, and this is a kind of midline transection through the location of the optic chiasm where the axons of, well, the nasal axons of the two eyes would be crossing the midline. And the first point to note is that sitting directly above the optic chiasm is this region of grey matter. It actually is part of the hypothalamus, and on the other side of the hypothalamus is the third ventricle. The third ventricle is the narrow slit between the left and right thalamus, and which sits also directly above the hypothalamus, and of course in your brain contains cerebrospinal fluid. So above, directly above the optic chiasm is the hypothalamus and the third ventricle, and just a short distance below is the pituitary gland, as indicated here. You may recall that the pituitary gland sits in a bony well called the pituitary or hypophyseal fossa. It's a bony well formed in part of the sphenoid bone. And from the centre of the chiasm here to the centre of the pituitary gland here is a distance of only about 2 centimetres or 20 millimetres. So a very short distance below the centre of the optic chiasm is the centre of the pituitary gland. So we have these major structures sitting directly above and directly below the chiasm, but there are also some really quite large non-neural tissues situated just lateral to the chiasm on both the right and left sides, and that's indicated in this other picture here. Just to give you a little bit of orientation, again we have the stumps of the optic nerves here. They're passing through the optic canal at the back of the orbit. The optic chiasm would be here. It would be an X-shaped region here, because this blob that you see is the pituitary gland situated underneath the chiasm. But the point about this picture is to show you what's just lateral to the chiasm on both the right and left sides. And one of the things which is immediately lateral is this thing labelled CS, which stands for the cavernous sinus. I think I mentioned something about the cavernous sinus to you before, but just as a reminder, the cavernous sinus is a network of small veins and arteries. I mean, some of those arteries are actually going to supply your pituitary gland. Some of the veins have really interesting names like the superior and inferior ophthalmic veins, which in fact have recently drained the entire contents of your orbit. So there's a kind of a network of veins and small arteries that form the sinus. And the sinus itself is surrounded by a bag of connective tissue. So that's one thing, the cavernous sinus. There's another thing as well, though, and it's indicated here, ICA, which stands for the internal carotid artery. The internal carotid artery, of course, is a really big artery that supplies blood to most of the front part of your brain, but it also passes up through the cavernous sinus before it gives off its very first intracranial branch, which is the ophthalmic artery, which enters the optic canal and branches to supply blood to all of the tissues of your eye and all the tissues of your orbit. So there's a really big artery, the internal carotid, that's running right next to the chiasm on either side laterally. Also surrounding the chiasm, in fact, is an arterial circle called the Circle of Willis, which we talked about in the first year. It's not just the internal carotid, but all of these other arteries. You see, once again, we see the stumps of the two optic nerves. This is the X-shaped region of the chiasm. And you can see there's some pretty hefty arteries all the way around here. You've got bits of the middle cerebral, bits of the anterior cerebral. You've got bits of the posterior and anterior communicatings, and bits of the posterior cerebral artery. So a large number of quite large arteries that surround the chiasm and form that anastomosing circle called the Circle of Willis. Actually, that turns out to be really useful because the different arteries that are components of the circle of Willis actually have branches which supply different parts of the optic pathway. So, for example, inside your orbit, the intraorbital part of the optic nerve is actually supplied directly by perforating branches of the ophthalmic artery. Where the optic nerve leaves the orbit through the optic canal, so that's the intracanalicular bit, that's supplied by short branches that come from the anterior cerebral and the anterior communicating arteries. Further back, as the two nerves come together and meet at the chiasm, the axons there are supplied by short branches of the posterior cerebral and posterior communicating arteries. So you can see there's a kind of, we're developing an anterior-posterior sequence here. And then the optic tract, and in fact also the lateral geniculate nucleus, are supplied by more branches of the posterior cerebral artery. They're called the perforating thalamic branches. Okay, so that's kind of set up some of the anatomy, and now I just want to talk briefly about the consequences of damage to axons in the vicinity of the chiasm. So we're going to talk about a couple of possible, and indeed a couple of them not that uncommon causes of compression of the axons that have come from the retina as they're passing through the chiasm. Just to make it clear, when axons are compressed, they don't convey action potentials any longer, so they effectively stop working. There are a couple of sort of major causes of compression of the chiasm right at the midline, right smack at the midline, that just damages the nasal axons that have come from both the left and right eyes. The classic one, of course, is a pituitary tumour, otherwise known as an adenoma. Adenomas are catch-all terms for tumours that appear in glands. They're typically benign, so they grow very slowly. But a pituitary tumour can only grow in an upwards direction to hit the optic chiasm from below. And here's another cause of compression of these nasal axons: raised pressure in the third ventricle, for example, in an adult person who's suffering from hydrocephalus. That can push the hypothalamus downwards and therefore damage the nasal axons from the two eyes from above. And if those types of damage are particularly severe, then the patient will have their nasal axons completely transected from both their eyes, and they end up with a condition which is called bitemporal hemianopsia. Bi because it's both eyes, temporal because it's the temporal fields of the two eyes that are affected, because it's the temporal fields that the nasal axons are looking at. Hemi because it's the half fields that have gone, and anopsia because the patient can't see them. But you can also get damage of the optic chiasm from the side, and this would just affect the temporal axons and only from one eye. So for example, you can get expansions of the cavernous sinus, perhaps as a consequence of a thrombosis in the veins or the arteries there. There's also a particular granulomatous bacterial disorder, Toulouse-Hunt syndrome, in which the cavernous sinus becomes inflamed, and it can also expand. And if the cavernous sinus expands towards the midline, it will hit the temporal axons on the lateral edge of the optic chiasm, the temporal axons coming from the eye on the same side. And another cause of damage to those axons would be an outpocketing, that is otherwise known as an aneurysm, a ballooning of the wall of the internal carotid artery. And if that balloon were to move towards the midline, again it could compress the axons on the lateral edge of the chiasm that have come from the temporal retina of the same eye. And that condition leads to a visual field loss which has another name. It's called uninasal hemianopia. Uni because it's only one eye, nasal because it's the nasal field of the eye which is affected, because it's the temporal axons that are damaged, hemi because it's half the field, and anopia because the patient can't see it. Remember, anopia and anopsia mean the same thing, can't see. The S is entirely optional. I would indicate, or just mention in passing, that if you have an outpocketing of the internal carotid artery due to an aneurysm, which is causing a patient a unilateral nasal hemianopia, I would consider that to be the least of their worries. I mean, I'd be a little bit more concerned about the D word if I were that patient. You remember the D word is dead. Because if their internal carotid artery aneurysm were to burst, that is unfortunately the likely outcome. You can also get damage to the chiasm, actually the nerve and the tract, pretty much anywhere along its course as a result of meningiomas. These are, again, benign tumours which affect the arachnoid layer of the meninges, and the whole of the optic pathways, once they leave the eye, the nerve, the chiasm and the tract have the meninges around them. And of course you can get visual field losses as a consequence of loss of blood supply, blockages or hemorrhages. I would like to mention, however, this is the reality of bitemporal hemianopia, and I've put the hemi in inverted commas here, because this is actually how a patient with a pituitary adenoma would more likely present at the early stages of their condition. And in this early stage of the condition, it's only the nasal axons from the inferior part of the nasal retina have been affected. See, the pituitary gland sits down here, and the pituitary gland's got a tumour in it, it's growing up, and the first axons it hits, okay, they're from the nasal retina, but from the inferior parts of the nasal retina, the inferior parts only. The superior parts of the nasal retina are up here, higher up in the chiasm. So as a consequence, in fact, the reality of the initial presentation of a patient who's got a pituitary adenoma is going to be just superior temporal visual field losses in the left and right eyes, which relate to the compression of the nasal inferior axons. If the tumour grows any further and is allowed to grow and become a macroadenoma, and a macroadenoma would be a tumour of at least 2 centimetres in diameter, and you remember that's the distance from the centre of the pituitary gland to the centre of the chiasm. If it gets to be 2 centimetres or more, then it will crush all of the axons or break all of the axons in the chiasm, and then the visual field loss will extend down here into the inferior temporal parts of the visual field, and then the patient will end up with a proper bitemporal hemianopia. I mean, the point here, of course, is, when tumours are growing from the pituitary gland initially, they grow slowly. And the second point is that they don't necessarily grow directly towards the midline, because this, again, is part of the reality of the early stages of bitemporal hemianopia. You see that the visual field loss is not the same in the left and right eyes. It's incongruent. There's a much bigger loss of the superior temporal field here in the right eye compared to the left eye. And why will that be? Well, when tumours grow out of the pituitary gland, they don't go, I've got to aim directly for the midline of the chiasm so that I produce, a bitemporal hemianopia. No, I mean, this tumour in this particular patient happens to be growing slightly more towards the right than it is towards the left. And as a consequence of that, the loss of visual field in the right eye is more severe at this stage than it is in the left. Okay. I think it's time just to take a little bit of a pause, and then I'll talk more about the geniculocortical system. Right, okay. So I showed you in the previous lecture a cross-section through the lateral geniculate nucleus, and this is it again. And I pointed out that the lateral geniculate nucleus comprises 6 separate layers of neurons. There are 2 down here at the bottom, layers 1 and 2, containing quite large neurons. These are known as the magnocellular layers of the lateral geniculate nucleus. And then there are 4 obvious layers above, 3, 4, 5 and 6, which are the parvocellular layers of the LGN. But here's another piece of information about the organisation of the lateral geniculate nucleus. We know that this nucleus receives input from the left and right eyes, from the opposite eye and from the eye on the same side. And in fact, the 6 layers of the LGN are shared equally between the 2 eyes. Specifically, nasal axons that have come from the opposite eye terminate in layers 1, 4 and 6 of the lateral geniculate nucleus, whereas axons that have come from the temporal retina of the eye on the same side, that's the ipsilateral eye, terminate in layers 2, 3 and 5 of the lateral geniculate nucleus. So here are the numbers of the layers, and here are the parts of the retina that send their information to each one. 3 of them receive crossed input, and 3 of them receive uncrossed input, but from the opposite half of the visual field. In fact, what this means is that each of the 6 layers of the lateral geniculate nucleus contains its own independent map of the opposite hemifield. So there are 6 maps of the opposite half of your visual field in your lateral geniculate nucleus. 3 of them from the nasal retina in layers 1, 4 and 6, and 3 of them from the temporal retina in layers 2, 3 and 5. And those maps are actually stacked on top of each other, a little bit like a sort of giant mega club sandwich, and all the maps are in alignment with each other. Now the neurons in each of those 6 layers give rise to axons of their own, and they're going to send information to the next level to the top level of the pathway, at the top of the food chain, which is the primary visual cortex. So, in other words, another function of the lateral geniculate nucleus is to organise the retinal inputs from the 2 eyes into these separate alternating maps of the opposite hemifield, one in each layer, before relaying the information onto V1 on the same, that is, ipsilateral side of the brain. And the axons that arise from LGN neurons in each of those 6 layers travel first in the internal capsule, in fact in its very posterior portion called the posterior limb, and then into the large optic radiations to reach area V1. And this is the geniculocortical pathway. Here we can see the lateral geniculate nucleus here on one side of the brain, here's the LGN on the other. And these bundles that you see are millions and millions of axons that have come from neurons in the lateral geniculate nucleus. They enter first into this white matter pathway here, which is part of the internal capsule, before fanning out here to form the optic radiations running around the side of this space here, actually quite close to the first part of the radiations, which is called the inferior horn of the lateral ventricle, which in your brain contains CSF. And then the radiations continue back here and end up here in the primary visual cortex, mainly in the medial surface of the occipital lobe. I've put, by the way, the inferior horn of the lateral ventricle in red because there's a warning here. And it relates to something I've told you right at the beginning of the lecture about 2 components of the optic radiations, which I'm now going to talk about for a few minutes. Okay, so this is this picture here in this slide. You see here is an expanded view of this region of the real tissue, and it shows this large cavity, which in a living brain is filled with CSF, so it's part of the ventricular system, and how it causes a problem to a particular group of axons that leave the LGN. Not to these ones here, which are indicated in purple. These are axons that are coming from the medial half of the lateral geniculate nucleus. And it so happens that the medial half of the LGN represents the lower visual quadrant. That's the lower part of the opposite hemifield, okay? So that half a hemifield is a quadrant. So the lower half of the hemifield, the lower quadrant, is represented by these axons that take a pretty direct route just over the top of the inferior horn of the lateral ventricle to get into the optic radiations very directly. But the axons coming from the lateral half of the lateral geniculate nucleus, which represents the upper visual quadrant, they are confronted by a problem. Their problem is that what they want to do is to grow directly across the inferior horn of the lateral ventricle, but they can't. Axons can't navigate through fluid, and so they are forced to do something that they don't really want to do, which is initially to travel in an anterior direction like this, completely opposite to that where the primary visual cortex is, and then form this kind of really obvious loop before turning around and then joining with the rest of the direct purple ones to end up in V1 cortex. So this is a figure which tries to sort of emphasize how there are these two components to the optic radiations. And again, I'm using the same color coding of purple and green. The purple is the direct, that don't have a problem with the ventricle, and the green are the ones that do. So here we're looking at the left side of the brain. Here's the left lateral geniculate nucleus, and here are the axons in purple coming straight out, joining the optic radiations, travelling underneath this, which is the parietal lobe, and ending up in the upper bank of the calcarine sulcus. And here are the axons that are coming out of the lateral side of the LGN in green, having to do this loop around the inferior horn of the lateral ventricle, just underneath the temporal lobe here, before joining the main bunch of the radiations and ending up in the lower bank of the calcarine sulcus. In fact, these axons that form the loop underneath the temporal lobe have a name. Those axons are called the axons of Meyer's loop. And there's something else also to point out here now. Remember, the purple axons represent the lower visual quadrant, and they end up in the upper bank of the calcarine sulcus, whereas the green axons represent the upper visual quadrant, and they end up in the lower bank of the calcarine sulcus. So if the lower quadrant is in the upper bank and the upper quadrant is in the lower bank, that means that the map of the hemifield is actually upside down in the primary visual cortex. That is, the hemifield map in area V1 is inverted, and as we'll see, it's also distorted. And we're going to look at these two features in relation to the right primary visual cortex here and its representation of the left half of the visual field. So this line here, this dotted line, corresponds to the vertical meridian. That's the vertical line that splits the left half of your visual field from the right, which isn't shown here. There's also another line that's running here at 90 degrees to it, this one here. This is called the horizontal meridian. Horizontal meridian. And it corresponds pretty much to your horizon. So, say you're standing on a beach looking out to sea, and you're looking at the level of the sea. Where the sky meets the sea, that would be your horizon. And that region of the visual field would correspond to this line here, the horizontal meridian. It looks at the horizon. So, as we can see before, again here, we've got this purple area here, which corresponds to the lower visual quadrant. That's the quadrant of the visual field below the horizontal meridian. And that's represented in the upper bank of the calcarine sulcus via the direct component of the radiations. And here's the upper visual quadrant in green, represented by the Meyer's loop component of the radiations ending up in the lower bank of the calcarine sulcus. So the map of the visual field is upside down or inverted from upper to lower. Another point to make, of course, is that the calcarine sulcus itself has a depth. It has a grey matter at the very base of the sulcus called the fundus, and it's within the fundus of the calcarine sulcus that the representation of the horizontal meridian between the lower and upper quadrants is found. So the horizontal meridian is represented right in the fundus of the sulcus. The second point is that the map of the hemifield in V1 is distorted. Particularly, there is an over-representation of the fovea and macula, and that's indicated here. You see this little sort of half circle? That corresponds to the very central part of your visual field, the fovea and macula, as I've said before, about 15 degrees, well, it would be 7.5 degrees just on this half side. And this dark region here of fovea and macular representation is found at the posterior end of the calcarine sulcus here. Whereas the more anterior, more peripheral part of your visual field here is represented here at the most anterior part of your calcarine sulcus. In fact, where the calcarine sulcus joins this sulcus here, which is the parietal occipital. So there is a centre to peripheral representation of your hemifield from posterior to anterior along the banks, the upper and lower banks and fundus of the calcarine sulcus. So centre here posteriorly and periphery here anteriorly. Notice also that this dark region corresponding to the fovea and macula is represented in a much larger part of the primary visual cortex, a larger part of the banks of the calcarine sulcus than the periphery here anteriorly. In fact, it's been reported that between 66 and 75% of the posterior part of area V1 is devoted to over-representing the fovea macula for central vision, with the peripheral parts of your visual field, which is really quite big. See all this stuff here? That's your visual periphery, and it's really enormous compared to this tiny little bit of the centre. That's all crammed into this tiny little area right at the anterior pole of V1 next to the parietal occipital sulcus. Right. The blood supplies to V1 cortex and the radiations come from two major cerebral arteries. They're the middle and posterior cerebrals. In fact, the middle cerebral artery supplies the geniculocortical axons in the internal capsule and in the very first part of the radiations. When the axons are in the last part, underneath the occipital lobe, in this region here, they're supplied by the posterior cerebral artery. And the posterior cerebral artery has a number of branches. I think there are 4 major ones. Here's one with an interesting name. It's called the calcarine artery. And guess what? This terminal branch of the posterior cerebral supplies the tissues in the upper and lower banks and indeed depths of the calcarine sulcus. So it's the calcarine artery which is the main branch of the PCA that supplies V1. I would point out that there's also quite another large branch here. It's called the posterior temporal artery. It also supplies quite a large part of the occipital lobe. Okay. So those are the blood supplies to V1. And now we're going to come on and look at some classic visual field losses associated with localized lesions or damage to different parts of the primary visual pathway. This picture is available in most, sort of classic textbooks, in chapters in books which are entitled, Human Anatomy and Physiology. So what we're looking at here is the primary visual pathways, and we've got 5 lesions, A to E, and these are the consequences, the visual field losses that result from each of these lesions. Okay, so this is the visual field of the left eye with its temporal and nasal fields, and this is the visual field of the right eye with its nasal and temporal fields. Remember, the nasal field of the right eye is pretty much the same region as the temporal field of the left eye, and that's the left hemifield. Okay, so lesion A is pretty straightforward. It's a complete transection of all the axons in the right optic nerve, and lo and behold, the patient wouldn't be able to see anything at all in their right eye. So they'd lose both the nasal and temporal fields in their right eye, but they'd be able to see perfectly well through their left eye. Lesion B is a classic midline lesion, which has damaged just the nasal axons coming from the right and left eyes. For example, the type of damage that you would expect from a really large pituitary adenoma, and that has caused this visual field loss, that is bitemporal hemianopia. Lesion C has knocked out and damaged all of the axons running in the optic tract, i.e. just behind the optic chiasm, on the right side of the brain. So the right optic tract is completely gone, and therefore neither the left nor right eyes are reporting information to the lateral geniculate nucleus, and the lateral geniculate nucleus therefore not to the right visual cortex. As a consequence of that, there is no information coming from the temporal retina of the right eye to the cortex, and no information from the nasal retina of the left eye, and therefore the patient can't see anything in their opposite hemifield. They will have this type of visual field loss, which is left hemianopia. That would also be true if they had lost the lateral geniculate nucleus on the right, or if they've got a complete lesion that totally wiped out V1 on the right. All of those lesions would lead to a left hemianopia. And the reason for that is because, the axons from the nasal retina have crossed the midline and the temporal axons haven't. So post-chiasmatically, beyond the chiasm, the representation is always of the opposite hemifield. Lesion D is meant to be a lesion which has just taken out the Meyer's loop component of optic radiations and left the direct component intact. And you remember the Myers loop component takes information to the lower bank of the calcarine sulcus about the upper visual quadrants, and therefore it's just the upper quadrants, again this is the right side of the brain, it's just the upper quadrants of the left hemifield that are gone, as indicated here. So the nature of this deficit is a left-sided problem. It's a left upper quadrantenopsia. Finally, lesion E is meant to be damage to the whole of the primary visual cortex, but resulting from occlusion of some part of the posterior cerebral artery. You remember that's the artery that supplies blood to pretty much all of the occipital lobe. And it's on the right side of the brain, so you're going to get a left hemianopia, as I've indicated before. But in this case, there is sparing of the fovea and macula, as indicated by this little region here, where the patient can still see, even though the rest of their left hemifield has gone in both the left and right eyes. So this is left hemianopia with macular sparing. And this, in fact, is a fairly common phenomenon, this macular sparing, associated with vascular problems related to the posterior cerebral artery. And a question arises, how is it that you can get this macular sparing when the artery has got a problem? Well, there is a fairly sort of standard, I mean it's in some textbooks, a fairly standard explanation for the macular sparing, and it relates to this, that this region back here, right at the very posterior pole of the calcarine sulcus, where the fovea and macula are mainly represented, has a dual blood supply. It doesn't just get its blood directly from the calcarine artery, which of course is this terminal branch of the PCA, but there is also a terminal branch of the middle cerebral artery that travels back and supplies this posterior pole with blood as well. So if the calcarine artery gets blocked or ruptures, then this selective region back here at the posterior pole is still being perfused with blood from this terminal branch of the MCA, and hence the neurons there will survive, they'll still be working, and the representation of the macula will be spared. Now personally, I think that's a really unlikely explanation for macular sparing. And so I'm just kind of playing around a little bit here right at the end of the lecture. I just wanted to point this out to you. I don't know if anybody knows exactly what this is, but I can tell you it's a carotid angiogram. And what's happened with this person is that a needle has been inserted into a carotid artery in their neck, somewhere around about here, and a contrast medium has been injected into that carotid artery. And then fairly shortly afterwards, they've undergone an X-ray, and that contrast medium shows up all the blood vessels in their head and neck, which are direct branches of the carotid. In fact, this one over here, that's the first branch of the carotid. It's the external carotid that supplies blood to most of the tissues of your face. And this one here is the internal carotid artery, running up here inside the skull. It's doing a funny little squiggle here. See this funny little U-bend? Actually, it does that U-bend just before it enters this region here, which is the region of the cavernous sinus. And what you can see here is all the blood that is flowing here in the anterior cerebral and here in the middle cerebral arteries, which are the two terminal branches of the internal carotid. So this is all the blood in the middle cerebral artery, and this over here is the posterior pole of the occipital lobe, where you find the representation of the fovea and macula. Can you see a branch, a terminal branch of the middle cerebral artery wandering over here to selectively supply blood to the fovea macular representation of V1? Because I can't. Moreover, neither could the people who did the carotid angiogram, because they've chopped off the posterior pole of the occipital lobe because there's nothing there. I can tell you I've looked at quite a few carotid angiograms, and I've never seen this mythical branch of the MCA wandering across to supply blood to the posterior pole of the occipital lobe. And indeed, I mean, what would be the evolutionary pressure to do that? You know, why would a branch of the middle cerebral artery go, oh, I've got to wander over, miles away over here to supply the occipital lobe, just in case this person has an occlusion of their PCA, and then I can give them macular sparing when they're older. The picture on the right is a post-mortem brain from somebody who died as a consequence of a hemorrhage of the posterior communicating artery. Sorry, posterior cerebral artery, PCA. You see all the blood here? This is the occipital pole where the fovea and macula is represented. Looks pretty yucky to me. Anyway, in 1966, two neurologists, Smith and Richardson, got hold of 32 post-mortem brains, and they went to the trouble of looking to see whether there was a dual blood supply to the occipital lobe in any of those post-mortem tissues. They followed the various arteries in the brains, looking to see whether there were branches back there that would supply the fovea and macular representation. And they looked at 32 post-mortem brains and published the results in the American Journal of Ophthalmology. And of those 32, they found 2 in which there was a hint of a branch from the middle cerebral supplying the posterior pole. 2 out of 32 is less than 10%. Whereas they found that in 16 of the post-mortem brains that they looked at, there was a branch of this one, the posterior temporal artery, which is a branch of the PCA that also supplied this region here where the fovea and macula are represented. And the truth of the matter is that most people who survive a posterior cerebral artery occlusion, in fact, it's only the calcarine branch which has become occluded. If the whole of the PCA goes, it's usually the D word. So it's only the calcarine artery that is affected when you get macular sparing. And when you get macular sparing, it's nearly always a consequence of a dual blood supply, but from the intact posterior temporal branch of the PCA, not from the middle cerebral artery at all. Okay, so this lecture finishes with a sample MCQ, which is kind of generally about the macula. The macular representation in area V1 is spared following posterior cerebral artery occlusion because it has another blood supply from the middle cerebral artery. Well, I don't believe that's generally true, but it is stated that that's the case in textbooks. So, unfortunately, that answer is true. Okay? If it says it in a textbook, and you've read a textbook, then I have to say it's true. And as you'll see, when you look at B, C, and D, they're wrong. You know, the macula isn't in the nasal retina. The macula itself doesn't cover all that 66 to 75% of the retinal surface. It's a much smaller fraction. And this is the question which is also important. Is specifically associated with axons coursing through Meyer's loop? No, I just wanted to make this point clear. Meyer's loop, which is this green one, and represents the upper visual quadrant, it also has the representation of the upper part of the fovea and macula, with the lower part of the fovea and macula being part of the lower visual quadrant that runs in the more direct component to the upper bank of the calcarine sulcus. Okay, well, I hope that was all abundantly clear, and if it wasn't, don't worry, because we're going to do some more on the visual field losses and visual field representations in this pathway in the tutorial, which is currently available to you on Moodle. And as you'll see, there are also some video recordings there that go through some of the visual field losses in slightly more detail than I've had the opportunity to do in this last hour. Okay.